Hello students, this video is going to be talking about complement, and it's going to be more of a procedural type of thing. I'm going to go from beginning to end, which is the opposite way that I did in class, at least this year that I'm recording this video. So hopefully this helps out. I have a lot of color coding going on, so once again, hopefully this will work out to help you step through complement. If we're looking at it from broad strokes, we have about 4 specific steps that happen for complement to happen. We start with activation, and there's 3 different varieties of activation, and I'm going to color code these. All of these are color coded later on, in case you wanted to actually care. Then we make a C3 convertase, we then make a C5 convertase, and then we pop the hole. And when I mean popping the hole, I mean popping the cell by making a hole. Activation, we have 3 different varieties of activation. We have the classical pathway, we have the mannose or the MASP pathway. Sometimes this is also called the lectin pathway, or the MBL pathway. It has a lot of different names, but still the same type of thing. And then we have the alternative pathway, which, as I said in class, this is actually going to be the most common. With the classical pathway, we start out with an antibody. So rather than having anything else, we have our antibody that is going to be bound to an antigen. And when it is bound to an antigen, likely on the surface of some sort of bacteria or something else like that, C1 comes along. And C1 actually has multiple subunits that are associated with it. Those are called QRS. Don't know why they're not ABC, but C1 QRS are going to bind to this antibody-antigen complex. That, as a broader thing, so this whole thing can act as a way of cleaving C2 into C2A and C2B, and C4 into C4A and C4B. This is one of those places where complement protein names have changed to make it easier. So depending on when you're looking, anything before about 2018 or so is going to flip-flop the C2, the A's and the B's. I believe that originally they did it based on size, not function. So, but C2B, so always remember B's mean business and A's go away. C2B and C4B are going to come together and make a C3 convertase. So this is a C3 convertase. And the C3 convertases are always going to cleave C3 into C3A and C3B. So that's what the convertases do. They split C3. So once again, classical, we have the starting point, which is antibody binding to the antigen. That is going to interact with C1, which will then, that whole thing will act as an enzyme to cleave C2 and C4 into C2A, C2B, and C4A, C4B. The B's mean business, and they're going to make a C3 convertase. The A's go away. And then they keep on doing whatever they'll do. We'll talk about what they do actually later on in the video. The mannose-MASP-MBL pathway is actually going to look very similar to this. Instead, we have mannose binding lectin, which is going to be binding to something on the surface of our bacteria, once again. So here's our bacteria. We have things on the surface that it is going to bind to, and that MBL will be doing the binding. MASP 1 and 2 are going to bind to that, and that complex, and really it depends on whether it's going to be MASP 1 or MASP 2, but both of them work together. But the MBL MASP complex is going to do the same type of thing as our C1 did and split our C2A and C2B and C4A, C4B. The B's once again get together and make a C3 convertase. The A's go away. Okay, so that should look very similar to this because it's pretty much the same. We have the same players. The starting point is different. The alternative pathway, we're still going to make a C3 convertase, but instead what's going to happen is that C3 will autolyse. it'll auto-hydrolyze. So there will be an H2O that'll be lost in the process. The A goes away. The B is going to do a couple other things. So B is going to actually bind to this other thing. We have factor D, which will bind to factor B and split factor B into BA and BB. Ooh, that's really similar colors in my mind. And if it's not, and one's brown and one's red, then once again, I am colorblind. In any case, the BB is going to bind to that C3B and make C3B B B. Okay, the capitalization is important because this capital B comes from factor B. Factor B, subunit B. And this C3B BBB acts as a C3 convertase. Our C3 convertase is going to be one of two different types. We either have a C4B2B or a C3B-BB. Both of these are going to be our C3 convertases and are going to split C3A into C3 into C3A and C3B. Once again, the C3A is going to go away. The C3B is going to work as our C5 convertase, bind to that C3 convertase, and then that is going to be our C5 convertase. So this C5 convertase is this complex of C3B added on to our C3 convertase. That means we either have two different types, once again, C4B, 2B, 3B, or C3B, BB, 3B. In any case, whatever it is, it's going to split C5A, which will go away, and C5B, which will go and do business. The C5B acts as the party planner, essentially, to bring all of the other complement proteins together. They have a party and make a hole in the membrane. And that hole in the membrane is largely going to be comprised of a number of these other different types of C9 proteins that are going to go around. C6, 7, and 8 are going to help stabilize it in the membrane. And then we have this movement of everything from the inside of the cell, out of the cell, through our hole in the membrane. We have our MAC formation. Now, about those A's, they all go away, and they're going to do a number of different types of things. They're going to activate immune cells in both the innate response and the T cells. They cause vasodilation, they activate macrophages, they cause inflammation, and they can also play a role in clotting. Most of the clotting is actually going to be more of that MASP. After they're done cutting things up, then they go on to help with clotting. But some of those A's are also going to take a role in that. So that's essentially everything that you end up doing through this whole process. Hopefully that helped. If it doesn't, I have a couple other videos, and just let me know. Okay. Bye.